The bee haemolymph is collected using dedicated in-house kits with a reduced invasive effect on the bees. Once collected, the haemolymph tubes are briefly centrifuged. For proteomics, the individual haemolymphs are pooled by 5 for each of the experimental conditions. The pools are organized according to the multi-molecular mass fingerprints. The pools are dried under vacuum for 30 minutes. Once dried, the pool is reconstituted in 20 microliters of the RapidGest reagent used to enhance enzymatic digestion of proteins. At this level, the pH of the samples should be between 7 and 8. Then, a volume of 2 microliters of a reducing agent is added to break the disulfide bonds present in some proteins. The reaction occurs in the dark for 45 minutes at 56 degrees Celsius. Afterwards, the samples are quickly centrifuged. Then, 4 microliters of an alkylating reagent are added to prevent cysteine from forming disulfide bridges, a second step necessary to facilitate the enzymatic digestion of the proteins. The reaction takes place in the dark for 45 minutes at room temperature. After this, the samples are again quickly centrifuged. For enzymatic digestion, trypsin is used as it generates short peptides easily detected by mass spectrometry, and the samples are incubated overnight at 37 degrees Celsius. After the overnight incubation, the samples are acidified and incubated for an additional 45 minutes at 37 degrees Celsius to stop the reaction. Then the samples are centrifuged at 15,000 G for 20 minutes, and the supernatant is transferred into a specific HPLC vial. The sample is automatically injected, and the products of the digest are analyzed by reverse phase HPLC coupled online to a high-resolution mass spectrometer. Following the mass spectrometry analysis, the Proteome Discoverer software is used to identify and quantify the proteins in a pool and to draw informative graphs. Using the OmicsBox software, the biological processes and the molecular functions of the dysregulated proteins are evidenced, and, more interestingly, the software allows us to identify the dysregulated biological pathways and localize the involved proteins. With Cytoscape, protein pathway networks are generated and analyzed in response to the stressors. These findings are the pillars to creating the 3 molecular databases on Apis, Bomis, and Osmia. All these studies are valued through scientific publications and international communications.